Hello viewers, today we are going to learn about immunoglobulin classes and their biological functions. As you know, there are 5 major classes of antibody molecules: IgG, IgD, IgE, IgA and IgM, and all these provide us protection from different microorganisms entering from different body sites, right? And you can also see that IgA is present here in a dimeric form, and IgM you can see a pentameric form. Actually, the case is that IgA can form dimer, trimer, tetramer, while IgM is either present as a monomeric form or a pentameric form. So let us discuss about each and every antibody molecules in a bit detail. So you can see here an IgG molecule having 2 light chains and 2 heavy chains. The proper structure that we did understand in previous lecture, that is a variable region and a constant region in the light chain, and 1 variable domain and 3 or 4 constant domains can be there in case of other classes. So this IgG is the most abundant uh, antibody molecule in our serum, and that is about 80% of the antibodies are do belong to this class IgG. And as we discussed while discussing different types and subtypes of antibody molecules, that IgG has different subtypes depending on the different kinds of gamma chain. So there are 4 kinds of subtypes, or I would say subclasses, depending on different kinds of gamma chain: IgG1, IgG2, 3 and 4. And the most abundant one in out of these 4 is IgG1, and out of these 4, IgG1, 3 and 4 readily crosses the placenta and provides protection to the during the gestation period to the embryo. And and also another feature of these antibody molecules is that they effectively activate complement system. Okay, and Ig3 is most capable in doing that, and it also mediate opsonization, that is antibody mediated phagocytosis by macrophages. So IgG2 is most capable in mediating opsonization. Okay, as I told you that these IgG is the only class that can cross the placenta. If you see the you know how does this provide passive immunity to newborns, you see here this is a timeline. The first half just before birth is showing the gestation period, and then the postnatal period of the newborn. You can see here the dark or the complete line shows the maternal IgG. In the newborn, after about 2 months, this IgG levels rises very quickly and reaches to its maximum. And while at about 6 months of or so during the gestation period, newborn starts making its own antibody, and that antibody is IgM. So the first antibody made in the newborn is IgM, right? Or first antibody made in case of adults also in an immune response is IgM. Okay, and then later other classes of or other subtypes are expressed. Then in newborn, a newborn starts making its own IgG, infant IgG, and later it starts producing other antibodies like IgA, E, and D. And after birth, maternal immunoglobulin IgG starts declining. And these are the 4 subclasses or subtypes of IgG: IG1, IG2, IG3, and IG4. You see how what is the difference, basic differences in their hinge region, number of the disulfide bonds, the length of the hinge region here is varying in these all molecules. IgG, you see this, the hinge region is the longest in the IG3. Okay, and the serum concentration, you can easily remember that this the same order, IgG1 is most abundant, and then their concentration gradually declines in the serum. So now you know that these IG4 IgG classes differ in their hinge region and in the length and the number of the disulfide bonds that they have. Here you are seeing the pentameric form of the IgM. Uh, about 5 to 10% of the serum IgG are IgM. Mostly when it is in membrane bound form, it is a monomeric antibody. Once it is secreted in the serum, it forms pentamer. Okay, and as we saw just a second before, few seconds before that, this is the first antibody found by the neonates, and also this is the first antibody during the primary response against any antigen. Okay, so whenever we are launching an immune response for the first time against an antigen, it will be the one, or the IgM will be the one, to be produced first of all. And this is also very efficient in agglutination, because the valency of this molecule is very high. See here we have 5 antibody molecules, so we have 10 antigen binding sites, so it is definitely more capable in agglutinating, as well as complement fixation. Complement fixation is actually a technique which is used to determine the presence or absence of antigens in any sample. We will discuss this technique later. Now this J chain allows the IgM to transport across the epithelium. Okay, so where in case of IgA also we see this J chain, and also in case of IgM. So this is the component of the oligomer that helps in the transport of this molecule across the epithelium. So that's why this antibody molecule is found in the secretions, body secretions, because this passes through the gastrointestinal, or respiratory, or you know, gen, you know urinogenital epithelium. So that's why this is found in the secretions. Here you are seeing the dimer of the IgA joined by the J chain. Okay, this forms of to 10 to 15% of the serum IgG. Okay, serum Ig or immunoglobulins, and it can form, as I told you earlier, also monomer, dimer, trimers, and tetramers. And it is the main predominant antibodies in external secretions like breast milk, saliva, tears, mucus or bronchial, urinogenitary tract or digestive tract. Here should be G, sorry for the mistake. And movement of the IgA across the epithelium layer is termed as transcytosis. Again, the J chain will be helping in the transcytosis. When IgA gets secreted into different body secretions, we find a secretory component associated with this. This structure you are seeing is a secretory component that we find associated with the IgA when it gets secreted out in the different body secretions we just mentioned. Okay, this secretory IgA, dimeric or tetrameric IgA, provides a very important line of defense against different bacteria like Salmonella, Vibrio, Neisseria, and also against certain viruses like polio, influenza, reoviruses. Right. So, because it neutralizes their antigens at the site of exposure itself, so it is quite important. Now, how this secretory component gets associated with this dimer? Let's see that. In the submucosal layer, many plasma cells, millions of plasma cells, actively secrete dimeric IgA, and this dimeric IgA has its receptor on the basal side of the epithelial lining, and this is a poly Ig receptor because it has multiple immunoglobulin domains. So this dimeric Ig along with the J chain, actually the J chain interacts with the receptor, and once this dimer gets bound to this poly Ig receptor, it gets endocytosed. So this is a kind of receptor mediated endocytosis, and this is this vesicle travels to the luminal side of the epithelium cell and fuses with the plasma membrane, and enzymatic cleavage of the receptor, see, this is the complete receptor, but once it is about to secrete the IgA into the lumen, the enzymatic cleavage, proteolytic cleavage of this receptor occurs, and this poly Ig domain gets remained associated with the dimeric IgA. So that's how we see a secretory component which is of poly Ig nature into the associated with the Ig molecule, and you will be surprised that we secrete about 5 to 15 grams of the IgA molecule in our intestine every day. So it's a huge amount, and you can Also imagine the importance of this secretory IgA in neutralizing different antigens entering through our gut. This whole process of endocyte secretion, endocytosis, and traversing of this vesicle to the luminal side of the epithelial cells, and transport of this IgA dimer into the lumen, this whole process from here to here, right, it is known as transcytosis, because it, in a way, it is traversing this epithelial cell, and the IgA dimer is getting transported out of the through the epithelial cells towards the lumen. So transcytosis, this process is known as transcytosis. IgE antibody, this E antigen, sorry. In case of IgE, this E terms come from the E antigen of the ragweed pollen. Actually, it was Prudesnes and Kushner in 1921 who indicated towards the presence of a serum component that was responsible for allergic reactions, and they were studying allergic reactions against the ragweed pollen, and they this duo only give the PK reactions or wheel and flare reactions. You must be knowing these are very well popular now to test whether a person has allergy towards a certain antigen or not. And it was K&T Shizaka in 1966 who identified the IgE subtype or isotype of the antibodies. It is really have high biological activity, and even in the least concentrations of in the serum, it can launch a very potent biological effect or response. It mediates immediate hypersensitivity reactions also, which we'll discuss later, and it is also responsible for the symptoms like hay fever, asthma, hives, and anaphylactic shock. So this is really important antibody in case of allergies. The antibody IgG is found in about 2% concentration of total serum Ig, and together with IgM, it is a major membrane bound Ig on the mature B cells, and also it is thought to function in the activation of B cells. The IgD has been found to bind to basophils and mast cells and activate these cells to produce antimicrobial factors, okay, which participates in respiratory immune defense in humans. And it is also found to stimulate basophils to release B cell homeostatic factors. So in a way, this IgD is probably involved in allergic reactions and activation of B cells and even homeostasis of the B cells. So viewers, thank you for watching this video. We will meet in our next video with more updates on biological functions of these immunoglobulins. Thank you for watching. Keep coming on to this channel for more videos. Do subscribe, like, and share. Thank you.